Ladies and gentlemen, good afternoon. This is going to be a very brief Unbreaking Science episode. I just want to point everyone to this stunning development right here. The SARS-CoV-2 RNA reverse transcribed and integrated in the human genome. This actually means that we may have an explanation for the prolonged apparent recurrence of PCR testing in patients after recovery, even though they don't have any further symptoms. The significance for this for managing the outbreak, the epidemic, the pandemic, whatever you want to call it, is immense. If the SARS-CoV-2 RNA genome is validated to have reverse transcribed itself and integrated into the human genome, then that means there are individuals walking around that express this PCR detectable nucleotide sequence. The authors are Zhang et al. This is on the BioRx archive. It is not yet peer-reviewed. So I want to just, for a very quick moment, say that if that study validates and it's replicated, then RT-PCR testing for coronavirus is all but done. What does that mean? That means that we're going to have to do good old-fashioned diagnosis where we culture the virus and make sure that the virus is present. Even sequencing the nucleotide doesn't mean that it's not from genomic DNA. So this is an added layer of complexity. It's obviously very unwelcome. And I will put the links in the comments of the video. That's all that I have to say. Here it is again, SARS-CoV-2 RNA reverse transcribed and integrated into the human genome. Have a look, read the article yourself, and we'll be back later, possibly with more details.